Hi. So for this video, right, I will be going through the worksheet 1 of enzymes and let's begin, okay? So in this worksheet, the first question is actually linked to chapter 3, which is biological molecules. You are given all these shapes that shows you the the types of nutrients like glycerol, glucose and all, okay? And so the next question, the question is actually asking uh, what constitutes from the shape, what constitutes a fat molecule, what constitutes a sucrose, and what constitutes a protein with 4 amino acids. Okay, so I think for fat molecule, most of you should be able to get the shape right. It is actually the rectangle and the three jagged uh, shapes. Okay, so something like this. Please draw properly, of course, and please make sure that they are connected because it is a bigger molecule. Okay, so yeah, this one's slightly wrong. Okay, so something like this. All right. Now, sucrose, right, is made out of one glucose plus one fructose. Okay, so you are supposed to draw two shapes. I can't remember what are the two shapes, but if it's a square and something else, you must show the bond, so which is a line, and then the other one, okay, something like this. A protein molecule with 4 amino acids. Amino acids comes in many different types, and so the shapes given are normally when we give a symbolic shape, right? The shape is always of a huge range. Okay, so you just have to draw any 4 amino acids, and you just gotta link them up like what I have shown you in the sucrose molecule. So it can be I do not know what's the shape again. Okay, so yeah, it must be like a circle, a square, a triangle, or I don't know, a diamond. Okay, so the more important thing is that you have to show that they are being linked. Okay, so this is what this question is asking about. All right. The part two of this question gives you an example of they use the word a large organic molecule. Okay, they don't tell you what it is. It is made up of all these five squares, and this is inferred, right, as a carbohydrate. Okay, so part one, right, they ask you what food group this molecule belongs to, and the inference is more of a carbohydrate than a protein. Like I just said, protein is made out of many amino acids, yes, but the types of amino acids, right, compared to monosaccharides in carbohydrates, they are more diverse. And so normally for amino acids, we don't show them using a single type of shape. Okay? And so for this particular question, the best inference is carbohydrates. All right? Now, then we move on to the functions of this of a food group. In this case, the function is a carbohydrate. When you write functions, right, you have to write them in full. Okay, so I lifted this all from the notes, and you can see, right, for carbohydrates, we normally give examples because there are so many different types of carbohydrates. And my advice, right, for writing functions, right, is to write them fully and not to write them in shorter versions. Okay, so yeah, it's actually pretty straightforward. It's just that the problem is, of course, remembering, right? And also to choose the correct function for the correct context. So normally, right, I don't really use like the last one. Let's take a look at the last one, right? I don't normally use this as a function because it's so unique. I normally use the first few ones. I normally use the first four. Okay, so yeah, this is my advice. Question two is an enzyme question. Okay, it is an action, and this they tell you that this action of bursting balloons, right, is basically an enzyme in action. Okay, so yeah, question two is an enzyme question. So I have an inflated balloon over here. Okay, on one end, right, if I poke it with a sharp pin, the balloon will burst. And if I don't poke it with a sharp pin, I poke it with the other end of the pin, the balloon doesn't burst. Okay, the pin bounces back and the balloon remains inflated. So they ask you, right, these two actions, right, what does it actually mean in enzymes? Okay, because they are because it's split into one action and split into another action, right? the answers that you can think of, right, is also pretty restricted. So let's move on to the question first, okay? So the first one, right, asks you what is represented by the inflated balloon and what is represented by the fragment. Now, if the pin is poking properly, it means that this balloon is converted into this. It shows that there is a reaction, and the reaction is of course enzymes, right? So the pin that breaks the balloon is the enzyme. Okay, so the pin is the enzyme. The inflated balloon is before it is burst, right? So before it is burst, it is inferred as the substrate molecule. And after it's burst, right, it is inferred as the product molecule. Okay, so the pin is the enzyme, the inflated balloon is the substrate, and the burst fragments of the balloon, right, are the product molecules. All right, write the word molecules, by the way. Okay, so suggest one way, just one, which the figure accurately represents an enzyme correct control reaction. So over here, right, you have to look at the picture and then you have to figure out with respect to the figure what properties of enzymes are you trying to write. So there are only two suggested answer, I feel. First one, right, you can see that the pin doesn't change. Okay, and so the pin remains the same even after I poke even after I poke the balloon, and it shows that enzymes are chemically unchanged after a chemical reaction. Okay, the word chemical is important, so please remember to write chemical. You cannot write enzymes are unchanged. It has to be chemically unchanged. Okay, after a chemical reaction. This can be seen from the diagram. Okay, you can see over in the diagram, right, that if I use the blunt end of the pin and I poke the balloon doesn't burst, correct? Even though there is contact, the contact must be effective. So if you refer to the theory, right, effective collision, so the active site of the enzyme must fit into the substrate, then it will work. And so over in the diagram, right, in the figure, it actually represents that because there are two sides of it. And so these two are my suggested answer. Okay. Then they will always ask, right? Not always last. In this case, right, they will ask, why does the figure not accurately represent? That means, what does it not show? If you remember all the properties of an enzyme, which you should, you can actually write quite a lot. Okay, and these are my suggested answer, and I think they are all legit. Because the diagram doesn't shows you everything. Okay, so yeah, I'm not going to go through every single line, but you're looking at 6 examples of what the figure does not show for enzymes. Okay, let's move on to question 3. Question 3 is an experiment question. So I have 4 types of liquid over here. One liquid, right, is pepsin and hydrochloric acid. The other liquid is just pepsin alone. Third one is boiled pepsin. And fourth one is the control, which is water. Okay. So there are 2 clues over here. First clue is that the milk proteins are large molecules that makes milk cloudy. Okay. The second clue over here is pepsin is an enzyme that works well in acidic condition. This one you should be familiar by now. Pepsin breaks down proteins to polypeptides, and pepsin is found in the stomach, which is acidic. Okay, so these 4 liquids, right, are being introduced to proteins over here like this, and you can see, right, that this is the start and this is the end, of course, right? So the end, right, you can see that the clear areas for 1 is bigger. There is also clear area for 2, but 3 and 4 don't have. Okay, so you have to start to think what is the uh, why is it? All right, but before that, please remember this. Pepsin is an enzyme that breaks down proteins to polypeptides, right? So remember your chapter 5 and chapter 4, okay? Yep. So first question, suggest the clear areas around 1 and 2. So 1 and 2, right, there is breakdown. All right, so breakdown. So you can see pepsin chemically break down proteins into polypeptides. This is the enzyme, this is the substrate, and this is the product. Okay, you are not supposed to write amino acids because the breakdown has to be done by stages. If in the context, right, the petri dish is originally cloudy. So the reason why it is cloudy, right, is because proteins is a large molecule, and because protein is a large molecule, right, it blocks out light. If proteins are being broken down into smaller polypeptides, they will be more they will the molecules are actually smaller, and so the solution, right, or the solution or the mixture will become clearer. Okay, so this is the third mark because of this, and it's due to polypeptides being smaller in size. Okay, but this two is more important because this two is more theory. All right, so like I said, 3 marks. If you can get 2 marks, it's quite good already. Okay, part B, explain the difference in the results between 1 and 2. So 1 is bigger than 2, right? So you must explain the difference. Okay, number 1, you realize that I can't remember which one is bigger anyway. Okay, but never mind. One of them has hydrochloric acid, and so hydrochloric acid provides an optimum pH for pepsin. Okay, optimum. Please remember the keywords in enzyme theory. So optimum pH refers to the best pH that pepsin can work in. And because pepsin can work so well in optimum pH under acidic condition, right? The one in cavity 1 can break down more proteins at a faster rate. And please remember to write to polypeptides. Okay, always write the products. Alright, whether it's 1 mark, 2 marks, or 3 marks. I think this is the last part of the worksheet. Okay, why is there no clear area around cavity 3? So cavity 3, right, you realize that the enzymes is void. But again, right, I have mentioned many times to you, please don't write down the word void. Please paraphrase what is void, okay? And so the keyword here is denature, not die, not destroy, okay? Please use the right keyword, and please write the paraphrase word for void, okay? There are two ways in which an enzyme can be denatured. Enzymes can be denatured by high temperatures, or enzymes can be denatured by extreme pH. So in this case, right, it is void, means it's 100 degrees Celsius, and so it is high temperature. Can? Okay. And lastly, cavity 4 is a control. And when you answer a control question, right, you cannot just write control full stop. Okay, you have to explain why, or you have to elaborate what does it mean by a control. So you can see, right, over here, even though you write control, you have to elaborate. And my suggested answer is to show that the clear areas is due to pepsin breaking down proteins. Okay, to show that the clear area is due to pepsin breaking down proteins. And so, yeah, in short, right, just try to elaborate on uh, a control in an experiment. Okay, so that's it. This is my take on enzyme worksheet 1. All right, I will talk about the other two, the enzyme worksheet 2 and the quiz in separate videos. All right, bye bye. Thank you.